Hello, welcome to QBI TV. I am Zelda Love, the Program Manager of the HIV Accessory and Regulatory Complex Center here at QBI UCSF. Today, we're excited to talk to Dr. Amber Smith, who recently completed the cryo-EM structure of the HIV nuclear export complex. Amber, welcome to QBI TV.
Hi, Zelda. Thanks for having me.
Some people believe that you cannot separate the science from the scientist, so we'll start our session by learning a little bit more about Amber. Amber, can you tell us a little bit more about your science training?
I guess I should start by saying that I began my love for science in high school. I got really close to a high school biology teacher, and so when it came time to go to college, I needed to put a proposed major for UCSC, and so my biology teacher suggested biology, so that's what I chose. And then I just continued along with that, and then I had this amazing opportunity to be part of a research program that gives access to underrepresented groups into labs. And so through that, I was able to do lab work as an undergrad. And that's when I realized that I love the lab setting and that it was kind of like my aha moment of this is where I belong. And so being a first generation college graduate, I had no idea about a PhD program. And so luckily the graduate mentor that I had while I did undergraduate research suggested that I apply to graduate school. And so then that led me to the University of Michigan to get my PhD.
Very awesome. Can you tell our audience a little bit more about your recent publication?
So our publication reveals the structure of the HIV-1 nuclear export complex. And this has been a complex that's been studied for, oh my gosh, almost 4 decades now. The cool part about being a member of HARC is that Michael Emmerman, who's also a PI in the group, he was the first person to identify Rev, which is the HIV, and the RNA portion, which is a viral RNA piece, also known as the RRE. And so Michael Emmerman, as a postdoc, identified this protein and this RNA as being essential in order to express HIV proteins for us to be able to study them and eventually, to use them for therapeutics. And so he identified the protein and the RNA, and from there on, the science, really took off. But it was a lot of biochemistry and cell biology, but the structural biology was really difficult. There were pieces solved using X-ray crystallography, but it wasn't until the advancements that have been made in cryo-EM that we were able to get this structure of the intact complex, including the human protein CRM1 and RAN-GTP, and then the HIV protein Rev and the viral RNA, the RRE. We used biochemistry to validate the observations that we saw within the structure. And so validating the binding sites of where Rev interacts with the human protein CRM1. Also, it was really exciting to see that the RNA interacts with the human protein CRM1. And this is essential because CRM1 is the major mammalian export receptor. And so not only are we revealing essential pieces to this critical step in the HIV life cycle, but we're also learning more about protein CRM1.
So it seems like it's almost like a multi-generational project that, as you mentioned, started with Michael Emmerman. And I'm sort of curious, does it seem like we had to wait until now to get the correct technology in place before we could really solve the structure?
I believe so. This was a very challenging target. The CRM1 in and of itself was a challenging target, although there had been X-ray crystal structures of the protein. The expression was done in bacterial methods, and it really needed mammalian, which large-scale mammalian expression has, come about or has been readily available to laboratories within like the last, 8 or 9 years. And so being able to express large amounts of CRM1 in mammalian cells was critical to getting this structure, in addition to the advancements in cryo-EM. So being able to collect large amounts of data and then functionalize grids and different data processing algorithms and programs. And so I worked on this project for 10 years. And so I don't know if we could have solved it any sooner.
Very awesome. So yeah, it seems like research takes a lot of sort of like problem solving. You have to be a little bit determined and resilient to, to start a project and see it through to the end to make it work. And I feel like this maybe tells me a little bit about yourself and about how you work in the lab. How do you approach lab work when things don't turn out the way you expect to?
My graduate studies played a part in this because the project that I worked on in graduate school was a project that, again, was structural, and it required getting a structure with substrates, which a lot of people had tried but couldn't. And so, it was kind of like this project, like we just need this one structure with the substrates to be able to elucidate the mechanism, the catalytic mechanism. And so I was able to achieve that. So kind of like closing the cap on things has kind of become my thing. And so when I came to UCSF as a postdoc, it was the same thing. Multiple people had worked on this. There's a lot of biochemistry, but how do all these pieces come together? It's really the support of my PIs, Yifan Chang and Alan Frankel. I mean, they just were so supportive throughout the way. And little advancements or things that I didn't really take as being accomplishments, they just made such a big deal about it. And so I think without their support just continuously pushing me forward, I wouldn't have felt successful to keep going.
That's pretty awesome. I think I think good mentorship is oftentimes sort of like we forget how important that is in our personal development. So thank you for saying that. So Amber, tell us why the general public should care about your discovery.
So the general public should care because HIV is still a global problem. There are hundreds of thousands that are being infected every day, and we have cases in every country around the world. And so this is still very much an important target. And in addition, viruses are, I'm not the first person to say this, but I love to use this when I present that viruses are the first biochemists of nature. In our quest to be able to develop therapeutics and to understand how viruses work, we tend to learn a lot about the host and the proteins that associate with the viral proteins. In order for us to understand how the viruses hijack them, we need to know how they behave in their normal situations. And so, like I said earlier, that this paper not only reveals information about the nuclear export of HIV viral transcripts, but it also revealed a lot about the human protein CRIM1. And CRIM1 is such an important protein for so many different diseases because of its role as a nuclear export receptor. And so we were able to really start to dive into how this protein that interacts with so many other proteins, including HIV, but not only HIV, there's numerous viruses that also interact with this protein. And so now we're also starting to decipher how this human protein interacts with all different types of proteins. And, this protein interacts with ribonucleoprotein complexes. So complexes made up of protein and RNA, which is similar to the HIV complex, but how this protein exports those. And so if we stop studying viruses, then we'll lose a lot of information that we've received from caring about helping people and curing disease.
Very, very awesome. Do you feel like this finding can maybe help lead to a drug discovery or a therapeutic that can maybe down the line benefit the general public?
Theoretically, yes, it should. Understanding protein structure and interfaces and how proteins touch each other. Obviously, if we disrupt those interfaces, then it would inhibit this step. And if we inhibited this step, HIV wouldn't be able to replicate. So long story short.
I guess in this case, what happens in the nucleus doesn't always stay in the nucleus, huh? So
true, very true.
It's a pretty bad joke, but anyways. All right. What does this publication and your discovery and completing this project mean to you personally?
It was just such a challenge, and it just seemed so impossible at times. And it just felt like I continuously made very tiny little steps until all of a sudden it was just this big leap, and then we had the structure. And then also it allowed me to pick up so many different techniques throughout this process. And so, not only do I consider myself a cryomicroscopist, but like also for very challenging targets, there's still a hole in electron microscopy for nucleic acid containing complexes. And so the techniques that I had to use in order to be able to see the RNA in this complex, I think will be beneficial to other scientists that are also studying ribonucleoprotein complexes.
Very, very awesome. Let's see, are you just sort of like thinking about your own career, looking ahead? Is this a type of work that you would like to continue to do in the future?
I love viruses. Like I said, I think that as a biochemist, studying viruses that are nature's own biochemists has always interested me. But ultimately, I just love proteins. So, and protein therapeutics is an emerging field where people are engineering and designing proteins that can now target different cell types and tissue types, and and then also the connection between viruses and cancer interests me too. But I would have to say the biggest theme of my interest lies within structural biology. I like to see how things work.
Very, very awesome. So before we close, I'm curious about maybe something you've learned that you might be able to pass on to a younger scientist who is watching this video. What advice would you give to a younger scientist, maybe a younger version of yourself 10 years ago or so, who's deciding to maybe embark on a career in science?
I have to say that I was really fortunate enough to be steered in the direction that I took with the mentors that I did. But I would say that that's the number one thing. You have to have supportive people around you. And so, make that the most serious aspect of deciding to continue on your career, is having very supportive mentors that believe in you until you believe in yourself. And so I think without that, I know without that, I wouldn't have completed this project.
Very awesome. Thank you for saying that. That was very insightful, I think. Do you have any closing remarks before we end the session?
The low-hanging fruit is not always it's not always the greatest feat. Don't give up on the challenging targets.
All right. Thank you for tuning in, and thank you, Amber, for being with us today. Please subscribe for more content like this from QBI TV.